And then the next speaker would be Dr. Ghazaleh Soleimani. And I think that Ghazaleh has some technical issues in terms of being connected. Nikki, we have the video from Ghazaleh as a backup plan. Can you play the video?
Yes, let me just set it up. All right, can you see my screen?
Perfectly well, yeah, please go ahead.
Hello everyone, I'm Ghazaleh Soleimani. Unfortunately, due to unstable internet connection in my location, I might not be able to have a live presentation, and I'm recording this video for the dose response webinar. Thank you, Hamid, for organizing these two fruitful webinars, and thank you for accepting this recorded video. In the next 10 minutes, I'll be going to talk about integrating electric field with brain responses to understand dose response relationship in brain stimulation studies. And here I want to be focused on MRI data because it can provide information about both the stimulation dose and the brain responses non-invasively. Based on structural MRI, we can create computational head models and electric field distribution patterns for each person. And based on functional MRI, we can provide information about brain responses to the applied brain stimulation technology. And integrating a computational head model as an indicator of received stimulation dose with fMRI data as an indicator of brain response could be informative. And we believe that this integration could be performed at least at 4 different levels: voxel level, regional level, cluster level, and network level. And we know that at each level we have different challenges. For example, for voxel-wise comparisons, both maps should be in the same space with the same resolution. Or for the regional level integration, we need to define seed regions appropriately. For example, we can use atlas-based parcellation, or we can define spherical regions around specific coordinates. But the size and location of these regions would be important for the integration of the head models and fMRI data. And we know that there are many other challenges. For example, there are many factors that may affect that could affect integration outcomes. For example, the accuracy of the electric field simulations and the accuracy of the fMRI analysis. And there are many unanswered questions in this field. For example, which measure should be extracted from each map? Do we need tangential or normal components of the electric field? Which one is better, resting state fMRI or task-based fMRI? Or if we want to extract a measure from a seed region, from a cluster, or a network, which one is better, median value, mean value, or maximum value? And many other questions in this field. And different approaches were used in previously published papers that used integration of the head models and fMRI data. For example, one of the first published paper in this field was conducted by Halko and colleagues in 2011, and they used regional level integration. And they reported a significant correlation between all of the voxels inside the seed region, predefined seed region, in computational head model and fMRI map for a single subject. In another study, voxel level integration was used for a group of participants. A sample standard head model was created as an indicator of a stimulation dose, and fMRI data were analyzed at the second level, and group level functional map was significantly correlated with the standard electric field distribution patterns at the voxel level. And at the network level, we couldn't find any previously published papers at the cluster level, but at the network level, for example, we found a study that reported significant correlations between maximum and median value of the current density inside the dorsolateral prefrontal cortex, and changes in the task-based functional connectivity in the working memory network in older adults. Despite the previous study, in this study, computational head models were generated for all of the participants instead of considering a sample standard head model. Network-based integration was also used for optimizing a stimulation dose by optimizing electrode placement over the scalp. We also explored all of these levels for determining the association between electric fields and changes in board signals based on one of our previously published TSFMRI database. In this In this database included 60 participants with methamphetamine use disorders, and in this parallel design trial, MRI data including structural MRI, resting state fMRI, and task-based fMRI were collected immediately before and after active or sham stimulation. And task-based fMRI was a standard drug query activity task. We generated computational head models for all of the participants, and for the voxel level integration, electric field maps resampled to have the same number of voxels with fMRI maps. And at the voxel level, we couldn't find any significant correlation between electric fields intensity and changes in board signals. And at the regional level, we couldn't find. We applied atlas-based parcellation, this atlas, which was BrainAtom atlas, was applied to the head model and fMRI map, and we extracted averaged electric field intensity and averaged changes in board signals from computational head models and fMRI maps. Again, we couldn't find any significant correlation between electric field intensity and changes in board signals across the population. And at the cluster level, we applied a second-level analysis of the general linear model, and we extracted active clusters while people were exposed to drug versus neutral cues. And then we extracted averaged electric field intensity and averaged changes in board signals from the active clusters. Again, we couldn't find any significant correlation between electric field and changes in board signals, but we found a significant effect of group at this level. For the network level integration, we calculated task-based functional connectivity inside the frontoparietal network, and we also extracted averaged electric field intensity from the frontopolar cortex as the brain region that received the maximum electric field across the population. And we found a significant positive correlation between electric field intensity and changes in task-based functional connectivity in the frontoparietal network, only in the active group. At the network level, we also checked resting state fMRI data. We calculated regional homogeneity during resting state, and we extracted average regional homogeneity from the large-scale brain networks. We also extracted average electric field intensity from these networks, and we found a significant negative correlation between averaged ReHo value in the default mode network and averaged electric field intensity in this network. And it seems that based on our result, network-based integration might be a better approach, and it makes sense because we have a diffuse current flow inside the brain, and and brain regions do not operate in isolation in different parts of the brain, works as a network. And although I only focused on integration of the head models and fMRI data based on correlation and regression analysis, this integration can be used for different purposes. For example, based on the information extracted from head models or fMRI data, we can classify the participants to responder and non-responders group, or we can validate our head models based on the fMRI data using the overlap between these two maps, and for many other purposes. We conducted systematic reviews to summarize all of the previously published papers in this field, and you can find our results in our OSF page right now. And we found that the number of studies which was published in electroconvulsive therapy, TMS or TS fields is limited. We have limited number of published studies in this field, and we are going to have a working group in the International Network of Neuromodulation Neuroimaging. And if you are interested in this field, and if you want to help us to have a consensus on integrating head models with fMRI data and to summarize the methodology for this integration, you can send an email to INTF network, and we would be happy to have more collaboration on this topic. Finally, if I want to summarize my presentation, I can say that I just wanted to highlight that collecting multiple modalities of MRI data can help us to understand dose-response relationship. Structural MRI can help us to create head models, and head models can be considered as an indicator of a stimulation dose. fMRI data or changes in board signals also can be considered as the brain response, and integrating the head models with fMRI can be considered as a measure of dose-response relationship. This integration can be performed at least at 4 different levels, and our results suggested that network-based integration might be a better approach, and our systematic reviews also highlighted that we have a few number of published papers in the field of integrating head models with fMRI data, and a consensus on dose-response computation discussing the parameter space for integrating head models with fMRI might be useful in the field of brain stimulation studies. Finally, I want to say thank you to all of my mentors, supervisors, and colleagues in different institutes, and thank you so much for your attention.